Hi everyone, today we are going to look at cell division. So in cell division itself, you have the division of the nucleus to produce two daughter cells. Now there's two types of cell division. I have got mitosis and meiosis. So in mitosis, it provides identical genetic materials, whereas in meiosis, you will have four non-identical haploid cells. So what it means by haploid, you can see that at the beginning it is diploid during fertilization, and at the end of it there's four identical haploid cells. So before we go into cell division, we need to understand what made these up. So essentially these are my chromosomes. So chromosomes, as you have learned under cells and structure, so it's made up of my chromatin. So chromatin, these are loose strands of DNA. So we got a couple of terms that look very identical, but they mean totally different things. So these chromatins, they are loose strands of DNA molecule. So when these loose strands of DNA molecule, they tighten up together, that's when I have got the condensed chromatin. So condensation of the chromatin is visualized under the microscope only during cell division. So if it is not condensed, means it is not during cell division, it won't be visible under a light microscope. So by microscope, we are saying the light microscope that we use in the labs. So this chromosome, I have got one chromosome and one chromatid and one centromere. So when this one chromosome undergoes cell division, I have got two chromatids, or rather cell duplication, I have got two chromatids. I still have one centromere. Okay, so see the difference between chromosomes and chromatid. So one chromosome, one centromere. So chromosome number is indicative of centromere number. Chromatid's number, on the other hand, is not indicative. Okay, so these two are identical. So these are what we call sister chromatids. So if I got, let's say, chromosome number 1 that duplicated to form two chromatids of chromosome number 1, these are homologous. Okay, on the other hand, chromosome number 2 and chromosome number 1, these are non-sister chromatids, so meaning to say they are not of the same family. So in humans, there are 46 chromosomes, 23 from the father, 23 from the mother, which is haploid, and then they fuse to become diploid. So X chromosome comes from the mother, Y chromosomes come from the father itself. Okay, so matching pairs of chromosomes are all homologous. So when you look at the karyotype over here, so these chromosomes, they are matching. So as you go down, it decreases in size, and of course, they are no longer homologous. So chromosome 11 and 12, they are not homologous, but chromosome 12, these two pairs are homologous. Okay, subsequently in meiosis, you have got pairing up of the homologous chromosomes. So as they pair up together, there can be synapsis. So synapsis is whereby there can be crossing over. So crossing over is whereby there is an exchange of genetic information. You can see the color change. So this exchange of genetic information happens when they cross over and forms a chiasma. So during that period, this will enable genetic variation, which is why none of us are 100% identical to the mother or 100% identical to the father. So types of cells that undergo mitosis or meiosis. Mitosis is for non-sex cells, so somatic cells, autosomal cells. Okay, meiosis, on the other hand, is for sex cells only, so gametes, aka also known as germ cells. So one thing here, it can be named different ways, but good to see the different terms over here.